Hello, everyone, and welcome to today's webinar on interrogating the SARS-CoV-2 specific immune response with single-cell resolution, end-to-end workflow. My name is Rita Isa, and I am the NanoSelect Technical Sales Representative for the Southwest. I will be your moderator today. First, I want to thank you for attending this virtual event. I hope you are staying healthy and safe. We're excited to join forces with Immudex and 10x Genomics in this informative webinar, and thank you for our speakers today, Dr. Sofina Taitano from Immudex, Dr. Nicole Jagnandan from NanoSelect, and Dr. Nicole Abreu from 10x Genomics. I am pleased to introduce our first speaker for today. Sofina Taitano received her PhD in immunology from the University of Michigan, studying regulatory B cells and their therapeutic potential in allergic disease. She then expanded her expertise to autoimmunity at the University of Colorado Anschutz Medical Center campus. Understanding how B-cell receptor editing during development can lead to autoreactive B cells and elucidating their potential role in lupus. Dr. Taitano is now an immune monitoring specialist at Immudex. Immudex developed Dexamer technology, enabled the monitoring, isolation, and in-depth profiling of antigen-specific T and B cells for immune system using fluorescence and next-generation sequencing methods. Dr. Taitano supports researchers in their utilization of Dexamer technology and manages business development in the Western North America. Before I hand the mic over to Dr. Taitano, we have a few housekeeping items to cover. First, today's webinar is being recorded and will be available on demand after this session. You will receive an email with a link to access it, and you can share it with your colleagues. If you have questions for the speakers, please feel free to type them in the Q&A box. Questions will be addressed at the end of the webinar. If we don't get your question during today's webinar, we'll be sure to follow up afterwards. So without further ado, I'd like to welcome our speaker, Dr. Sofina Taitano.
Thank you, Rita. Good morning, everyone, and thank you for joining us today to learn about COVID-19 immune monitoring solutions from Immudex, NanoSelect, and 10x Genomics. And I would first like to thank NanoSelect for hosting and our partner company, 10x Genomics, for the joint effort on this webinar. For today's talk, we'll start with a look into the viral immune response. Then we'll introduce how Dexamer and ClickMer technology allow you to detect antigen-specific T cells and B cells. And lastly, we'll talk about SARS-CoV-2 specific reagents for immune monitoring. So a really nice review came out a couple of weeks ago summarizing what we know about the immune response and infection of SARS-CoV-2. SARS-CoV-2 infects cells expressing the surface receptor ACE2 and the protease TMPRSS2. And these allow the virus to invade the cell. The active replication and release of the virus causes the host cell to undergo pyroptosis, which is an inflammatory type of cell death. Pyroptosis results in the release of damage-associated molecular patterns, or DAMPs, including ATP, host DNA, and ASC oligomers, and also the release of pathogen-associated molecular patterns, or PAMPs, such as viral RNA. These PAMPs and DAMPs are recognized by neighboring cells and alveolar macrophages, triggering their production of pro-inflammatory cytokines and chemokines. And these cytokines and chemokines attract monocytes, macrophages, and T cells to the site of infection, promoting further inflammation and establishing this pro-inflammatory feedback loop. In a healthy immune response, the initial inflammation attracts virus-specific CD8 T cells to the site of infection, where they can eliminate infected cells before the virus can spread. CD4 T cells are the conductors of the immune response, priming CD8 T cells and supporting the generation of neutralizing antibodies. These neutralizing antibodies can block viral infection, and alveolar macrophages recognize neutralized viruses and apoptotic cells and clear them by phagocytosis. So altogether, these processes lead to the clearance of the virus and minimal lung damage, resulting in recovery. In a defective immune response, the response may lead to further accumulation of immune cells in the lungs, causing overproduction of pro-inflammatory cytokines, which eventually damages the lung infrastructure. The resulting cytokine storm circulates through the blood to other organs, leading to multi-organ damage. So beyond controlling the virus, we also need to think about controlling the immune response, as disease severity is not only due to the virus, but also the inflammatory response of the immune system. In early studies using basic immunophenotyping of T cells, B cells, and NK cells from the blood of patients that has already begun to separate patient populations. So here, using blood samples taken upon admission to the hospital, patients with either moderate in gray or severe disease in blue have significantly decreased T cells, with patients that went on to develop severe disease having a greater reduction than moderate disease patients. When we look at predictors of mortality, low CD8 T cell counts in the blood is a strong predictor of developing severe disease and mortality, as you can see with that very significant p-value. Beyond being reduced in absolute number, CD8 T cells also show signs of being functionally exhausted. So blue here is patients with mild disease, and red is patients with severe disease. So CD8 T cells from mild and severe patients express the inhibitory receptor and exhaustion marker NKG2A, along with decreased cytokine and granzyme B production. As patients recovered, their CD8 T cell numbers began to be restored and the exhaustion markers reduced during this convalescent period. So these observations seem to agree with the important role of the T cell response, also seen in the previous SARS virus in 2003. With the previous SARS virus, a strong T cell response correlated significantly with higher neutralizing antibodies in convalescent patients. And in mouse model studies, it was also found that virus-specific T cell responses were necessary for viral clearance and protection. So T cells likely play an important role in the viral response during COVID-19, and biomarkers from the immune response could potentially help discriminate between patient populations in a predictive manner. So with the eagerness to reopen businesses and restart the economy, another important question that remains is whether naturally acquired immunity through infection with SARS-CoV-2 will protect from reinfection. And as infection with SARS-CoV-2 only began 6 to 7 months ago, there's no data on how long humoral and cellular immunity lasts. But since SARS-CoV-2 shares 79% sequence homology with the SARS-CoV virus from 2003, and they utilize a similar mechanism of entry, we can look at the longevity of the immune response to the previous SARS virus to speculate what we might expect with this novel SARS-CoV-2. So from follow-up studies of patients that recovered from the previous SARS virus, it was found that circulating SARS-specific antibodies peaked at about 4 months, which is circled, and then began to decline thereafter. In a 6-year follow-up study, SARS-specific IgG antibodies and memory B cells were undetectable in the blood of recovered patients. However, memory T cells were still detectable 6 years after disease onset. Now, whether the memory cells and or the antibodies would actually protect against reinfection is still unknown. And with so many unanswered questions and a large gradient of responses from asymptomatic to fatal, there's a need for extensive immune monitoring and profiling. And to assist in the efforts to understand the SARS-CoV-2 specific immune response, Immudex empowers researchers with our Dextramer technology to profile antigen-specific T cells and Clickmer technology to profile antigen-specific B cells. Our technology allows in-depth interrogation and monitoring of the antigen-specific immune response by flow cytometry, next-generation sequencing with our Decode Dextramer reagents using the HIT DNA barcode for bulk analysis, and single-cell next-generation sequencing with our Decode Dextramer reagents using the 10x Genomics compatible DNA barcode, which we'll go into a bit more in a few slides. So first, let's break down the Dextramer and how it helps you detect antigen-specific T cells. So Dextramers conjugate an optimal number of peptide MHC complexes and multiple fluorophores onto its flexible dextran backbone. And the flexibility of the dextran allows the Dextramer to wrap around the cells, utilizing all of those peptide MHC complexes without steric hindrance. This increased number of peptide MHC complexes enhances the sensitivity to enable detection of a wider range of T cell receptor affinities, and the additional fluorophores enhance resolution of the positive population. So you have a higher mean fluorescence intensity. So T cell receptors can bind to their MHC peptide complexes with different affinities, with low affinity T cell receptors being between 20 to 500 micromolar and higher affinity TCRs being around 0.1 to 10 micromolar. And the affinity of the T cell receptor is actually a poor estimate of the T cell response, with many examples of functionally relevant T cells being of low affinity. So being able to include even these low affinity T cells gives you an unbiased and comprehensive view of the response. The multimerization of the MHC peptide complexes onto the dextran backbone increases the avidity, which is particularly advantageous for these low affinity T cell receptors. And in fact, in a publication comparing tetramer and dextramer for the detection of high and low affinity T cell receptors, they show that staining with the dextramer allows detection of antigen-specific T cells with low affinity T cell receptors that tetramers cannot detect. And so this new level of sensitivity uncovers antigen-specific populations that could not previously be identified. On the other hand, for high affinity T cell receptors, both tetramer and dextramer can detect these antigen-specific populations, but dextramer staining shows better resolution of the positive population because of those multiple fluorophores that are bound to the backbone. Now, beyond detection by fluorescence, the 10x compatible Decode Dextramer technology adds a unique DNA barcode to the MHC Dextramer reagent. And this allows high throughput screening of multiple T cell specificities within the same sample. So each Decode Dextramer has a unique barcode encoding for the MHC peptide specificity it displays, a primer sequence for Illumina sequencing, a unique molecular identifier, and a capture sequence for the 10x gel bead oligo. Now let's take a look at how the Decode Dextramer fits into the 10x workflow. So first you would stain your sample with a panel of Decode Dextramer reagents, and then using the PE fluorophore that's conjugated to the Dextramer, you sort on the Dextramer positive cells. And so this is to enrich for the antigen-specific cells. And no matter how many specificities you're multiplexing, they will all be PE positive because it's the unique barcode on each Dextramer that will distinguish between the specificities in the end. And since antigen-specific cells are also a very low frequency, they can be like 1 in 10,000 cells, This enrichment step is crucial if you're trying to profile the antigen-specific population. So after sorting, now you're ready to load your sample onto the 10x Chromium system to be partitioned into single-cell reactions. And after library preparation and sequencing, you now get data on the antigen specificity, paired T-cell receptor sequence, cell surface protein expression, and the gene expression for every single cell. So to assist researchers, Immudex has curated a panel of SARS-CoV-2 specific dextramer reagents. And this panel includes 30 dextramer reagents with epitopes from the spike, nucleocapsid, and membrane proteins of SARS-CoV-2 and covers the 4 most common HLA alleles. The panel also comprises of 4 negative controls and 4 positive controls. So if you're interested in this panel, feel free to reach out to us. And also, if you're interested in CD4 T-cell responses, we also do have some reagents available related to COVID-19. And we are currently working on a second panel to put together more MHC class 2 dextramers. So now to talk about B cells, utilizing our CLICMER and our DNA barcoded decode dextramer create reagents, you can interrogate the SARS-CoV-2 specific B cells with that same high sensitivity and resolution that you get with our dextramer reagents. The CLICMER technology allows you to attach any biotinylated protein or peptide to the multiple streptavidin molecules conjugated to the backbone. And so in the case of COVID-19, you could use samples from infected or vaccinated individuals to help accelerate antibody discovery and epitope mapping, assess vaccine candidate selection, and stratify patient populations through immune monitoring. So using decode dextramer and decode dextramer create reagents to interrogate the SARS-CoV-2 immune response, you can help answer many of the remaining questions. For example, what role cross-reactive epitopes play in disease severity? So last week, a paper from the study group at the La Jolla Institute of Immunology found that 50% of their healthy controls had cross-reactive T cells to the SARS-CoV-2 virus. So what are those epitopes and do they correlate with protection? Can we use biomarkers from antigen-specific immune responses to stratify patient populations and answer questions about disease severity and potentially how the diversity of the HLA phenotypes may be affecting the responses? And lastly, monitoring the immune response can also help assess and guide development of vaccines and therapeutic candidates, potentially correlating or predicting response to treatment with the immune profile, as has been seen with immuno-oncology. So I'm going to end here, and feel free to reach out to our team if you are interested in the SARS-CoV-2 panel or discussing your project needs. And I just want to mention that we have many more human, mouse, and macaque MHC alleles available than the 4 that I mentioned today. And we take custom peptide and allele orders every day. So if you don't see what you need on the website, feel free to reach out to us and request a certain specificity. So thank you.
Thank you, Sofina, for a great presentation. Our next speaker is Nicole Jagnandan from NanoSelect. Nicole earned her PhD in microbiology from the University of California, Riverside. During Dr. Jagnandan's academic career, she studied viral immunology and host pathogen interactions by using a rhesus macaque animal model. Dr. Jagnandan had been working as an application scientist at NanoSelect Biomedical for over a year, and now where she plays an instrumental role in leading our single-cell genomics and T-cell application nodes.
Thank you, Rita. All right, so for my portion of the talk, I'm going to be focusing on the cell sorting step of the workflow and the importance of using a safe and gentle cell sorter when working with infectious cells. I will specifically be focusing on Oh, sorry. Can you guys see my screen?
Yes.
Okay, thank you. I will specifically be focusing on how cell sorting can be beneficial for COVID-19 research, why the Wolf cell sorter is most ideal for infectious disease testing, how cell sorting fits into a 10x genomics workflow, and lastly, how low pressure cell sorting can reduce background noise and improve sequencing results. So flow cytometry has played an instrumental role in studying the immune response to COVID-19. We now know that people who are infected with COVID-19 exhibit significant changes in their lymphocyte and macrophage population. We also know that this virus causes a severe inflammatory cytokine response, and this imbalance of the immune response is what drives the development of serious complications. Viruses rely on a host for survival. They hijack our cellular machinery in order to replicate, and they have evolved ways to evade the immune system. These mechanisms can be more easily figured out through cell sorting because this allows you to focus on a specific cell type. This allows us to better interpret disease prognosis, the antigenicity of viral proteins, and the immune response profiles. Sorting for a specific cell type before your downstream applications can also allow you to better understand the effects of cytokines and chemokines, as well as the kinetics of the humoral and T-cell specific response for discovery and the development of therapeutics and vaccines. And lastly, and what I'm going to be focusing on, is that cell sorting can improve the quality and reliability of your downstream assays, like in 10x genomics single cell sequencing. When preparing your sample for single cell sequencing, it's important to treat your cells as gently as possible. Things such as using wide bore pipette tips, avoiding excessive vortexing, using gentle centrifuge speeds, and using the most optimal buffer for your cell type are all important techniques you should use. You should also remove any debris, dead cells, or cellular aggregates from your sample. Not taking these precautions can lead to a clog during your gem generation step, degraded RNA, inaccurate sequencing results, and a significant loss in money. Fluorescently activated cell sorters can be a valuable tool in your sample prep for 10x genomics applications. Debris, dead cells, and sticky cells or doublets can all be removed with a cell sorter. And you can also isolate your target cell type using fluorescent antibodies. By doing this, you're going to have a higher percentage of healthier cells for your genomic library prep, which means you're going to have higher RNA quality and better sequencing results. However, there are some safety concerns because high pressure cell sorters can generate biohazardous aerosols. They can also cause shear stress, which can lead to cell damage and gene expression changes that can alter your sequencing results. So it's really important to use a gentle cell sorter. Which brings me to the Wolf cell sorting. The Wolf cell sorter, on the other hand, uses really low pressure to sort cells. It's also very small and can easily fit into a biosafety cabinet. The Wolf also uses sterile and disposable cartridges and generates zero aerosols, so you can feel more safe when handling infectious cells. And you don't have to worry about cross-contamination. The Wolf also comes with its own intuitive software, and setup and cleanup only takes a few minutes. And it's also much more affordable than other cell sorters, making it possible for each lab to have its own cell sorter. Conventional cell sorters that use a cuvette or a jet and air sorting mechanism can exert anywhere from 10 to 70 pounds per square inch of pressure of your cells on your cells, which causes a lot of shear stress on them. This also causes this decompression shock once the cells exit the nozzle. The WOOF emits just under 2 pounds per square inch of pressure on your cells, and there's no decompression shock once the cells exit out of the cartridge, which means your cells end up being much healthier and functional for your downstream assays. As I just mentioned, a significant advantage of our WOOF cell sorter is that the entire fluidics pathway and everything that the sample and sheath touch is disposable and sterile. This means that you don't have to worry about there being any cross-contamination between samples. This feature, along with the capability of using the WOOF in a biosafety cabinet, allow you to truly work in sterile conditions when working with infectious samples. So the whole sorting process occurs on a disposable microfluidic cartridge. After laser interrogation, this PZT here receives a voltage charge that causes it to bend or push or pull the cells into the right sorting junction. And here's a video of the sorting process using a high-speed camera with black dye, showing how the fluidic pathway changes as the PZT pushes or pulls. Here are the key components of the WOOF cell sorter. As you can see, everything is easily accessible through the front of the machine. The cartridge gets inserted here and the tubing gets threaded through these peristaltic pumps, which pump your sample and your sheath into the cartridge. As you can see, very little sheath is required for our machine. 50 mils of sheath is all you need for a full day's use, and this can be customized to whatever sorting buffer is most optimal for your cell type. Inside the WOOF, there's a 488 nanometer laser, which allows you to sort based on 5 parameters, forward and backside scatter, and 3 fluorescent channels. So you can use up to 3 fluorescent markers to identify your cell type. In the 10x Genomics workflow, the WOOF cell sorter can be used prior to loading your sample into the Chromium controller. During this step, you should isolate your target cells and remove any dead cells or aggregates. After sorting your cells, you would then concentrate your sample down to the 10x Chromium kit recommendations prior to loading your sample into the Chromium controller. From there, you would proceed with their library generation protocol and then sequence your samples. In order to validate the genomic benefits of using the WOOF cell sorter prior to loading your sample into the Chromium controller, PBMCs were isolated from human blood and then split into 2 samples. In one sample, 10,000 cells were spun down and then directly loaded into the Chromium system to serve as our unsorted sample. The remaining PBMCs were stained with a viability dye and ran through the WOOF for sorting. We then sorted for the live PBMC population by gating out the debris and dead cells using a viability dye and any doublets. 10,000 of these WOOF sorted cells were then spun down and loaded into the Chromium. Both of these samples were sequenced through the Illumina HiSeq 4000 and then ran through the Cell Ranger pipeline with the help of the UCSD Genomics Core. So the Cell Ranger summary produces several key metrics about the RNA and sequencing quality. The sequencing analysis for our 2 samples showed that there was almost 3 times more median genes per cell in our sorted sample compared to our unsorted sample. There was also more total genes detected in our sorted sample, and both of these results show a higher library complexity was achieved from sorting. There was also a lower percentage of reads that were not associated with a cell in our sorted sample, which is a more favorable result because this means that there was less ambient RNA, likely from dead cells. We then looked at the t-SNE plots generated from these two samples. So these t-SNE plots here are showing a heat map of the number of unique molecular identifiers or RNA transcripts in each cell. Blue meaning a lower amount of UMI counts and red meaning a higher amount. You can see that there is much more blue dots in the unsorted PBMCs compared to the sorted PBMCs. We then looked at the numbers and saw that there was almost 4 times more median UMI counts per cell. And this dramatic increase in UMI counts is likely due to the fact that there was less dead cells and fragmented RNA. There was also 2 more clusters of cells detected in the sorted sample, and the clusters and transcriptional profiles between each cell type in the sorted PBMCs, which was much more distinct because of the higher library complexity and reduced background noise. We then wanted to see how these results compared with other cell sorters. So we took some stem cells and split them into 3 samples. 1 sample served as our unsorted control, and the other 2 samples were stained with a viability dye and either sorted through the BD-FACS ARIA2 or the WOOF. Both samples were sorted at the same time to exclude dead cells and doublets. The ARIA sorted sample was sorted by the flow core at Sanford Burnham Prebys using a 100 micron nozzle and at 20 psi, compared to the 50 micron channel in the WOOF cartridge at 2 psi. 50,000 live stem cells were sorted on both machines, and then around 10,000 cells were loaded into the Chromium controller for library prep and sequencing. All 3 of these samples had cDNA libraries generated using the Chromium NextGem single-cell 3' kit, and sequencing was done on the Illumina NovaSeq. Data analysis for these samples was also run through the CellRanger pipeline. The sample quality metrics for these samples showed that there was a 3-fold increase in the WOOF sample and a 2-fold increase in the FACS ARIA2 sample in the number of median genes per cell, which again shows that sorting results in an increase in library complexity. We also saw an increase in the number of median UMI counts per cell, with the highest increase in the WOOF sample. Sorting also resulted in an increase in the percentage of reads that mapped to the transcriptome, which means that there was less fragmented RNA and contamination in the sorted samples. And so overall, these experiments show that sorting enables you to get more data per cell, and that this may be even more improved by sorting your samples with a gentle cell sorter such as the WOOF. In conclusion, due to the ability of the WOOF to be placed in a biosafety cabinet and the disposable sorting cartridges, the WOOF cell sorter is the most ideal for sorting infectious cells. Also, our experiments show that the quality of your sequencing results is directly dependent on how you prepare your sample. And finally, using the WOOF cell sorter for your 10x genomics applications can improve library complexity and reduce background noise from dead cells and debris while eliminating the risk of cross-contamination. And I would also like to say that the WOOF can be used for several other downstream applications. And the WOOF has been recently selected by the Human Immune Monitoring Center at Mount Sinai to help study COVID-19. And finally, if you'd like to learn more about cell sorting and COVID-19, we do have an informational resource page on our website. And that's all I have. So I'll now turn this over to Nicole Abreu from 10x Genomics. Thank you.
Thank you, Dr. Jagnandan, for a great informative presentation. And our next speaker is Dr. Nicole Abreu. Dr. Abreu earned her PhD and postdoc in microbiology from the University of California, Berkeley. There, she studied bacterial genetics with respect to cell biology and nutrient sharing in the microbiome. Afterwards, Dr. Abreu worked at Resilient Biotics, an Illumina accelerator startup company, before joining 10x Genomics. At 10x Genomics, Dr. Abreu enables customer science as a science and technology advisor for San Diego and Orange County. Dr. Abreu?
Perfect. Thanks, Rita. Are you guys able to see my screen?
Yes, we can see
Yes.
it.
Perfect. Thanks. So hi, everyone. Thank you for attending today's webinar. Also, thanks to my colleagues, Sophina and Nicole from Immudex and NanoSelect for their fantastic presentations. So today, I hope to share how 10x Genomics can be used to study the current COVID-19 pandemic. So unfortunately, the SARS-CoV-2 virus has reached all corners of the globe, and it's at the forefront of our minds right now as we've been gearing up to combat this disease. It's so encouraging for me to see how the tools that 10x Genomics have provided have enabled researchers to better understand this infectious disease. As the pandemic is ongoing, we've answered back by publishing research on this virus and how it impacts the host on a single cell level. I'll state that many of these publications are in preprint, but examples of such papers include research that have profiled cells from patients with varying degrees of COVID-19 severity, and have also begun to elucidate the host response to SARS-CoV-2. And another recent study has identified neutralizing antibodies for SARS-CoV-2 as well. So a lot of various researchers from across the globe have really been aggressively looking to use our tools to better understand what's going on in this pandemic. So as far as the types of research that we see are tools to enable researchers that are interested in studying the SARS-CoV-2 virus, I'd like to think of them as placing in two different buckets. One area where we can think of single cell and spatial solutions to answer such questions as analyzing single cells and tissues before, during, and after viral infection. Characterize the relationships between infection and the cellular phenotype that's present, study immune activation in response to infection, and identify the molecular and cellular mechanisms of infection. So a number of different tools that 10x Genomics provides, such as the single cell gene expression solution, single cell immune profiling solution, single cell attack solution, and spatial gene expression products, can enable answering such questions. Additionally, doing research in more of the vaccination and treatment space, we think that the single cell immune profiling solution is ideally poised to answer these types of questions, such as identifying antigen-specific immune responses to vaccines, find antibodies from patients that correlate with infection and might be protective potentially for the broader population, and assess antibody specificity for known or putative antigens. So for today's talk, I wanted to speak about some in-house data that we did here at 10x Genomics to identify some antigen-specific immune responses to vaccines. And so, as Sophina already eloquently and Nicole eloquently described, the immune response from a T-cell perspective is mediated through the interaction between a T-cell receptor and its cognate antigen. And what the 10x Genomics Immune Profiling solution enables is the capability to measure simultaneously all of these different aspects of this interaction, be it the gene expression profile of, in this example, a CD8 positive T cell, the clonotype of the T cell receptor that can interact with a particular antigen as well. So you're able to measure all of these metrics simultaneously from the same cell. In this example, we have a cytomegalovirus seropositive donor where we looked at different viral antigens such as Epstein-Barr virus, of course, cytomegalovirus, and a flu antigen. We were able to identify subpopulations of cells that responded to each of those individual antigens. And for this particular example, we have blown up the TCR clonotypes for that subpopulation. Of course, you'd get that from these others as well. And so how that occurs is essentially by supplying the 10x system with a flexible input of a single cell suspension. We can profile anywhere to hundreds or thousands of cells from an individual channel of a chip. That chip goes onto the Chromium controller, which enables our single cell gem generation. So that's how each of the individual cells become barcoded and labeled with a unique barcode that can map all of the transcripts and molecules from that cell to a particular barcode. Then we supply the reagents to generate the sequencing libraries so that they can be sequenced on a platform, a sequencing platform, and the robust analysis pipeline and visualization tools afterwards to visualize your data. So a little bit more granularity into how that occurs. So within the chip, you would apply your cells. In this case, these cartoon examples are cells that we've also labeled with antibodies that have oligonucleotide tags, or as Sofina mentioned earlier, the decode dextramer that has the antigen with its respective oligonucleotide tag. So these cells get applied onto the chip with 10x barcoded gel beads. So these are the 10x barcodes that I mentioned earlier, where an individual cell will get encapsulated in a gem and have its respective 10x barcode assigned to it. So that occurs by collecting the gems off of the chip and running a reverse transcription reaction that labels the cDNA with the respective 10x barcode. Then you would pool the sample and do your downstream library preparation in bulk, where each of the molecules that you have from each cell have their unique barcode. And as far as the output, what you would be able to get here in this schematic is information for each individual cell, their gene expression profile for each individual cell. In this case, cell 1 is a T cell, so you get the TCR clonotype of that cell. Cell 5000 is a B cell, so you would get the heavy and light chain information. And then a feature type of information. So in this case, a feature can be an antibody count, so a particular cell surface protein quantifiable metric, or in the case of the decode dextramer, you would identify the antigens through this enablement. So now to the study that I had mentioned that we performed in-house. So we were interested in understanding at a single cell level the response to an influenza vaccination. So what we did was we took 4 donors and we did 2 blood draws, one before and one 14 days after an influenza vaccination. And we studied 3 populations of cells from the blood, PBMCs, and enriched T cells and B cells. We took these 3 different populations and we added additional feature barcoding type features to each of them. So for instance, for the PBMC population, we added 84 antibodies So keep in mind that since you're using a oligonucleotide tag in this case, you can multiplex multiple different antibodies and not worry about spectral overlap. So 84 antibodies in this case for the PBMCs. For the T cells, we added a 19 antibody T cell panel, and then we included these decode dextramers, 10 different ones that were specific to either flu, the Epstein-Barr virus, or cytomegalovirus. And then for the B cells, we used 17 antibody B cell panel. We performed whole transcriptome gene expression on these samples, the immune repertoire profiling. So again, that's getting the paired chain information for T cell receptor or B cell receptor. And last but not least, the feature barcode capability, where we looked at both cell surface protein and antigen specificity. So again, I'm emphasizing that you can get all of this paired information at a single cell level. So what did this data look like? So in this particular study, we wanted to look at a very large number of cells. So we profiled almost 1.3 million single cells for this experiment. And just to get a sense for the throughput or the workflow aspect of getting data for this number of cells, this was generated in only 1 week, and it used 8 Chromium chips to do so. And as I mentioned earlier, these are the different metrics we were studying: gene expression, immune repertoire, cell surface protein, and antigen specificity from these cells. Here are the decode dextramer epitopes for the various viruses that we profiled. And as far as the data that we received afterwards, we were able to do different things, such as identify rare clonotypes of T cells that were present after vaccination, before and after vaccination. We were able to see B cells that were engaging in class switching after vaccination. We were able to identify subpopulations of different cell types, such as B cells, T cells, macrophages, monocytes, look at B cells that were re-entering memory, and also identify the subpopulation of B cells that were expanded in response to vaccination. So a lot of data all on one slide, but ultimately I hope that this highlights the power that you can get with only a week worth of work with 8 Chromium chips to really look at various layers of information from single cells to better understand the immune response to vaccination. And so, you know, we are of course very interested in helping our customers and researchers become enabled to tackle this research question for many reasons, right? And so we've got a lot of resources so far that we put on our website. That link there down below is where you would go to enable getting more information about what we have to help people with COVID-19 research. Such as publications and those preprints, like I mentioned at the beginning of the talk, and different resources for demonstrating the use of our solutions. There are webinars as well on this link, including a talk from Dr. Setliff at Vanderbilt University that used a tool to identify neutralizing antibodies to HIV infection using our approach. And also another webinar from our 10x Genomics staff scientist, Sarah Taylor, that looked at profiling the immune response to influenza vaccination. So please go ahead and check this out if you're interested. I myself am definitely here to help if anyone has any additional questions or would like to set up time to discuss your research question. I'm definitely here to help. Thank you for your time and attention. And with that, I'll turn it back over to Rita.
Thank you, Nicole, and thank you for all our speakers for amazing presentations. We will now start the Q&A session. If you haven't done so, please type questions into the Q&A box and we will answer them according to our time limitation. If we cannot answer your question now, we will follow up with you. So our first question seems to be for Immudex and maybe 10x as well. Are there any tools for studying specific T-cell responses in murine models for SARS-CoV-2?
Yeah, so this is Sofina from Immudex. We don't currently have any on the shelf, but we're surveying the literature and kind of keeping on top of what epitopes might be most pertinent for these murine models. And if you have any epitopes that you're interested in, feel free to reach out to us. We have a list of our MHC mouse alleles on our website, so you can take a look at that and then let us know what epitope you're interested in, and we can always do that as a custom peptide order.
Thank you. The second question is for NanoSelect. How long can you use the cartridges for, and are they reusable?
So the cartridges are good for 1 full days of use. They're not reusable. However, you don't have to change out your cartridge in between every sample. You can just do a flush in between samples if you want to.
Thank you. Another question for NanoSelect. What is the sorting speed of the Wolf?
So with the WOOF, our focus is really to be as gentle as possible on your cells. So our sorting speed is 300 cells per second, and it can do up to 2 populations at the same time. So it's definitely going to be fast enough for a lot of your downstream applications, such as 10x Genomics.
Thank you. A question for Immudex. Do you recommend using a negative control decode dextamer?
Yes, so negative controls should be used for every decode dextramer experiment. And so this helps you get the most clear data that you can. So when you use negative controls, it allows you to set a threshold for the background, and then you can get a really clear signal of which cells are actually antigen specific and antigen or epitope positive. So definitely negative controls are important, and we have a general negative control on our website, and then you can also use allele specific negative controls.
Another question for 10x Genomics. Are there differences in gene expression from cell to cell quantifiable?
Absolutely. So the benefit of doing single cell gene expression analysis is you will get individual cellular information about gene expression. So in short, you can definitely get cell-to-cell variability in gene expression quantifiably measured.
Another question for Immudex. How important is it to sort the antigen-specific cells before loading onto the Chromium controller?
Yeah, so that's a great question. So antigen-specific cells can be very rare and usually are, which can be maybe 1 in 10,000. And when you think about how many cells you can target per well on the Chromium trip, you can target about 10,000 cells. So if you are looking at an antigen-specific population and you don't sort, you might only get 1 antigen-specific cell out of all of those 10,000 cells you're targeting. So it's really important to sort and enrich for the antigen-specific population if your question is directly about that antigen-specific population.
Thank you. Another question for Immudex. Is it possible to trigger BCR cross-linking in B cells and activate them using the antigen-specific clicker?
Yeah, that's an interesting question. because it's higher avidity, that's potentially a possibility, but we don't have any data to support or disprove that. So that would have to be looked at experimentally, but I can see how that could definitely be a potential use of the CLICMER.
Thank you. This coming question is, I think, for 10x. How long can I hold or store my samples before loading onto the Chromium controller? Does it matter?
That is a very good question. So, you know, in general, we do recommend trying to run the samples on the Chromium controller as quickly as is possible, given the logistics of your experiment. We have seen some sample to sample type variability as far as how flexible you can be with that time frame, right? Because, you know, depending on the cell type, if they're left out for far too long, you can imagine that there can be some transcriptional changes occurring at a single cell level. So, you know, in short, I would say that many people do things like, for instance, for PBMCs, they'll freeze them. So you can generate a PBMC population, freeze that down so that you've got your pool of, let's say you want to run 4 at the same time or 8 at the same time, have those all frozen, and then thaw them all at the same time, quantify them, and load them onto the instrument. So there definitely are some experimental considerations to think of when you're deciding how to set up your experiment.
Thank you, Nicole. And due to the time, we're going to take our last question, and it is for Immudex. Thanks for your succinct presentation. Is it possible to provide you with a specific antigenetic epitope and get Dextamer or CLICMER conjugated to the epitope provided? How long would be a turnaround time per epitope? Or together, say, if we provide 20 epitopes?
Yeah, that's great. So definitely we take custom peptide orders every day. So I would say the best thing is to send over what your 20 different epitopes are, and then we can kind of look through them and let you know if they are actually a custom peptide order, or if somebody else has already kind of ordered that, then it's a standard order. So if we've already made it, it would take between 1 and 2 weeks to get delivered to you. And then if it's truly a custom order and we need to order the peptide, it'll take about 6 weeks because we have to get the peptide in, and then we do a lot of different quality controls. Yeah, so 1 to 2 weeks if it's standard, and then around 6 weeks if it's actually a custom order.
Thank you, Sofina. And with that, we will end the webinar series. Thank you again, everyone, for attending. I would like to take this moment to introduce you to the rest of the Southern California team here with NanoSelect and my partners in crime, Elena Bartakova and Jerome Karpiak, who are the field application scientists here. Thank you again for attending, and please stay safe and healthy.